Welcome to solverfy.
So how is a linkage map created? A linkage map, remember, is a map that tells you how different chromosomes in a, how different genes in a chromosomes are linked to each other. What is the order of genes in a chromosome? And you can actually get this by studying the process of recombination. Recombination is the process of homologous crossover during meiosis one. So in the production of gametes, you have a chromosome from the paternal side and a chromosome from the maternal side. They line up. There's some crossing over that happens, and then in the production of gametes, the gametes every single one of the four gametes. So remember, from meiosis, you go from one cell to four, and every single one of the cells is haploid. So at the end, you get four cells that each have one of these sister chromatids in their in their nucleus. So crossing over is the recombination of homologous chromosomes during meiosis one in the process of gamete formation. Good. Now, suppose that you have two genes of interest that you're looking at, and you want to find out where in the chromosomes those two genes are located. So let's hypothetically label these two genes A and B and say that A is closer to the center of the chromosome than B is. And let's say that if it is on the red chromosome, gene A is the recessive type, and if it is on the green one, it is the dominant type. So let's look at our case one here. Suppose that crossing over happens at this location. If crossing over happens at this location, this leg of the red chromosome will be not affected, but this leg now has the original recessive A, but it now has the dominant B, whereas this leg of the chromosome B or of the green chromosome has the recessive B and the dominant A that it used to have, but it picked up the recessive B from the red guy. So in case one, A is not recombined, whereas B is recombined. Now let's look at case two, where both of these guys are crossed over. So the crossing overs between the two chromosomes happens above the location of A. So in this instance, you will have this leg where you have the dominant A and B, and on this leg where you have the recessive A and B. So in this instance, both A and B are recombined. Okay, they are crossed over. And from these results right here, you can then make a call as to which chromosome or which gene is closer to the center of the chromosome. If we observe that any time that A is recombined, B is also recombined, which is case two, but that's not always the case for vice versa. So if B is recombined, that doesn't necessarily mean that A is recombined. That tells you that A sits closer to the centromere, to the center of the chromosome than B does. And now you have a relative map concerning genes A and B. And if you repeat this process with all of the genes in a chromosome, you can then build a relative map, a linkage map of all the genes in that chromosome. So let's look at our answer choices. Which of the following is true about linkage analysis? It is used to create a physical map. No, a physical map also tells you how much of a chromosome a specific gene takes. This doesn't tell you whether the gene is 1,000 nucleotides long or 10,000 nucleotides long. It only tells you the relative position. So no, it's not used to create a physical map. Choice B, it is based on the natural recombination process. Yes, that is correct. That's exactly what we described. Choice B is correct. Choice C, it involves the breaking and rejoining of DNA artificially. No, you don't need to do it artificially. This is a process that happens naturally in cells. And choice D requires radiation hybrid mapping, which is a method of artificially breaking DNA. So it's wrong for the same reason as C. Choice B is the correct answer.